welcome to our third online lecture. In the last online lecture, we talked about cell structure and function. We talked about different organelles. The one organelle we want to focus today is the cell membrane or the plasma membrane. We will talk about the structure and the function of the plasma membrane today. The plasma membrane separates the internal and external environment. Why? Because the external environment, they are unstable. They change a lot. They change all the time. So, but a cell requires stable environment so that the chemical reaction can consistently and repeatedly occur. Therefore, having a plasma membrane to separate the internal and external environment is necessary. The plasma membrane also regulates the in and out of different molecules. For example, you want nutrients such as glucose and oxygen to go into the cell, and you want a waste like carbon dioxide and urea to go out of the cell. And to maintain the constant stable environment inside the cell so that the chemical reaction can consistently and repeatedly occur, we call this homeostasis. We talked about homeostasis in the first lecture, in class face-to-face lecture, and I mentioned that homeostasis is a very important theme in physiology. And plasma membrane is made up of phospholipid bilayer. It has the hydrophilic, that means water-loving polar head, and this hydrophilic polar head is the phosphate group. And then we have the hydrophobic or water-fearing non-polar tails. Those are the lipid tails. So polar head, hydrophilic polar head is the phosphate group. Hydrophobic non-polar head, they are the lipid tails. You have, usually it's like one polar head with two lipid tails. And then you have the cholesterol. Cholesterol controls the excess fluidity, meaning that it controls how easy, how soft the cell membrane is. And you have protein going through the cell membrane. For example, you have something called peripheral protein. Peripheral protein attached only on one side of the membrane. It can be the outside of the membrane or it can be inside of the membrane. They only attach to one side of the membrane. But integral protein, they go through the membrane. Integral protein can also be called transmembrane protein because it goes through the membrane from one side to another side. It protrudes from both sides, or it can only one side embedded in the membrane. The membrane protein, they can move laterally in the phospholipid bilayer. You can consider the phospholipid membrane is like a ocean or a sea or ocean. Things can flow in the ocean or the sea. It's like the protein can flow in the phospholipid bilayer cell membrane. The fluidity or how easy to flow is controlled by the cholesterol. So protein, you have different types of protein. You have something called glycolipids. So glycolipids, they can attach to the carbohydrate. So the protein, you have different antenna or different molecule can be attached to it. One is called glycolipid. Glycol meaning the sugar, lipid is the lipid. So it's a lipid sugar molecule that can attach to the protein. And then you also have glycoprotein. Glycoprotein, glycoprotein as a name is the sugar molecule and some amino acid chain. And they are also can attach to the protein. And it says that here carbohydrate. What it means is that the carbohydrate chain, that means the sugar chain, it can have a lipid or protein branch to it. So the protein, the integral or peripheral protein, they can have a carbohydrate chain attached to it, and the carbohydrate chain can be glycolipid or glycoproteins. So you can see in here, there's a better depiction. So you have the integral protein and you have the peripheral protein. And then all these, you can have glycoprotein, phospholipid attached to the protein. What do they for? They are for identifying the cell. Our immune system can recognize this glycoprotein or phospholipid to identify whether the cell belong to you or belong to the invader. So it is one way to tell, for the immune system to tell between whether it is your own tissue or foreign tissue. And also on the red blood cell, you also have glycoprotein. The glycoprotein on the red blood cell determines the blood type. And then you have channel protein. Some of the protein on the membrane, they work as a channel. So what is channel? For example, ion, like sodium ion or potassium ion, they are charged molecule. They have positive charge with it, so they cannot go through the phospholipid bilayer because you have the non-polar lipid tail in the middle. So the ion, the charged ion, cannot go through the non-polar tail. In this case, what happens is that we have this protein channel that go through the cell membrane. It is part of the integral protein, and so that the ion can go through the integral protein, we call it channel protein, to avoid the hydrophobic tail. Carrier proteins, they help to move a larger molecule across the membrane. For example, glucose. Although glucose is non-polar, but it is very large. It is so large that it cannot go through the cell membrane. So what happens is that it needs a carrier protein, also an integral protein, to push the glucose into the cell. Cell recognition protein. Cell recognition protein, as I mentioned before, glycoprotein is one of the examples. It helps your immune system to tell whether or not this cell belongs to you or this cell is a foreign invader. And then receptor protein, they bind to a signaling molecule. There are many types of signaling molecule. The two common types of signaling molecule is the hormone. Hormone is a signaling molecule. So when hormone binds to the receptor protein on the cell surface, then hormone can bring changes to the cell. Or neurotransmitter. Neurotransmitter is also a signaling molecule that can bind to the receptor protein on the nerve cell or neuron. Enzymatic protein. Enzymatic protein, as the name suggested, it is a protein that can speed up the reaction of certain chemical reaction. And it's said that at the end of the electron transport chain, there is an enzymatic protein. We call it ATP synthase. ATP synthase is the example of enzymatic protein at the end of the cellular respiration that can make over 30 ATP for one glucose molecule. We will talk a lot more about cellular respiration later. So this is just a repeat of what we just said. Channel protein, it can transport charged ion, in this case chloride ion, to go through the cell membrane. Carrier protein, as I told you that glucose is a large nonpolar molecule. Because it is large, even though it is nonpolar, it cannot go through the membrane. Then you have this carrier that can carry the glucose to go through the cell membrane. Cell recognition protein, they are the glycoprotein. In here, it gives you an important example. It's called MHC, major histocompatibility complex. It is important because you will learn about this again in physiology. It is for your body to tell whether or not it is your foreign MHC or your self MHC. So if your body determines it's foreign MHC, then your white blood cell will attack. Then your receptor protein, I told you that a hormone or neurotransmitter can bind to it so that it can trigger some downstream cascade, downstream chemical reaction to bring changes to the cell. And then enzymatic protein. The example is the ATP synthase. In here, it gives you another one, it's called adenylate cyclase, which the cholera bacteria will release toxin to inhibit or interfere with this adenylate cyclase protein to affect the downstream signaling.